So cancer is very different from all of these other genetic disorders. For most of the genetic disorders, we have one genetic variant or a few genetic variants that are co-inherited. So every person that is predisposed to that particular disease because of that genetic difference, all of these individuals have the exact same genetic difference. So we can actually go and study systematically the relatively small number of differences in the human genome between individuals and simply characterize them fully, figure out how they act and how they manifest themselves in all of the individuals that carry these genetic differences. Cancer is fundamentally different because every single cancer cell accumulates a different set of mutations. So that basically means that we cannot simply build a catalog of all mutations because these mutations can be anywhere in the genome. So that catalog would include every single one of the 3.2 billion nucleotides of the human genome and all three ways in which they can change. So what we instead need to do is actually understand how to actually interpret de novo mutations, mutations that have only arisen once, perhaps in the history of all cancer, that disrupt that one cell in that one individual. So when we're talking about personalized medicine, when we're talking about individualized medicine, when we're talking about precision medicine, that basically means going to cancer and understanding for every single tumor in every single patient and every single cell in that tumor, how has that circuitry changed? And the reason why this is so important in cancer is because it's not simply inherited mutations. These are de novo mutations. These are new changes, new lesions that are actually under the process of evolution, that are undergoing rapid evolution within the body of that individual and that are constantly changing. So a tumor that you see today is the result of the accumulation of these mutational changes as well as epigenomic changes. So what our lab is doing right now is actually focusing on a subset of tumor cell types and going after the circuitry of these cells, going after the differences between individuals who carry the same tumor type, understanding the epigenomic landscape of these differences and translating this landscape into, number one, patient stratification, to understand how are different patients similar to each other at the circuitry level, and number two, how their cells respond to treatment, how their cells respond to drugs. So we're building three-dimensional models of these cancers and we're perturbing these models with different therapeutics and seeing which ones of those actually work inside those cells in order to then apply them directly to the patient and tune the treatment to the specific patient, to the specific tumor class, and to the specific cells of that tumor. Our work is not focused on any one disease area. We actually span multiple disease areas because of the foundational work in understanding disease mechanism, which actually spans across different symptoms. So one big area that we've been working on is Alzheimer's disease. So for a long time, people have thought that Alzheimer's disease has, of course, everything to do with the brain, and specifically the neurons in our brain that are degenerating. What our research has shown is that instead, the majority of the action, at least on the genetic side, appears to be happening on the immune cells that are surrounding these neurons. They're known as microglial cells. And the vast majority of genetic signals that predispose us to Alzheimer's disease are in fact sitting and acting right within these immune cells, which suddenly shifts the focus, the therapeutic focus, from intervening at the neuron level to intervening at the immune cell level, which might actually make it more accessible to new therapeutics. The other disease area that we're working on is obesity. So our work found that the strongest genetic association with obesity is in fact not perturbing the brain and appetite the way that people had previously thought, but instead is affecting a master switch inside our own fat cells that decides whether these fat cells will store our excess calories as fat, as lipids, inside our white fat cells, or whether they will actually burn these excess calories away as heat in brown fat cells or beige fat cells. And what we found is that we can actually manipulate that switch. We can flip it back and forth and take fat storing cells and turn them into fat burning cells. And that has tremendous implications for actually addressing the obesity epidemic in our planet, which has only shown increasing rates of obesity as the excess calories in our diet became abundant throughout the human population. And that switch is actually disrupted in about 40% of Europeans. And that basically means that by manipulating that circuitry, not just in the European population, but across the world population, we can perhaps bring a third pillar towards addressing obesity, in addition to, of course, healthy eating and a healthy lifestyle, to basically help our own body's metabolism in burning away those excess calories.